the technical uh, on the technical topic of applications in toxicology and chemical risk assessment for transcriptomics. We'll then, at the end, shift gears a bit and have a discussion or a presentation on taking a strategic approach to your career development. And next slide, please. First, I want to just mention that all of this would not be possible without the support of our GEMS Board of Directors. First in the list after myself is Katie Bailey, and she will be the one who will be leading this meeting. And so with that, I will turn it over to Katie, and it's all your show.
Okay, great. Thank you, George. Hello, everyone, and welcome to our virtual 2021 spring meeting. And really thank you for joining us. Hopefully this will be our last virtual meeting for a while. So here's the agenda for today. As you can see, we have a total of 4 presentations. As George mentioned, there's 3 that are covering transcriptomics and how it can be used for chemical risk assessment. So that's sort of the scientific talks, followed by a presentation on career development. So as you can see, we'll have 2 talks followed by a short break, and then 2 additional talks before we adjourn at 3:30. So before we get started, let's just go over a few logistical items. First, please keep your microphone muted during the presentations. If you're unmuted and we detect some background noise, we'll go ahead and mute you, okay? So the presentations are about 20 minutes each, which should allow for some time for questions at the end of each talk within the 30 minutes allotted. If you have a question, please raise your hand using the reaction feature in the Zoom at the bottom of your screen, or you can type a question in the chat. Any typed questions in the chat that we can't get to during the meeting will be forwarded on to the relevant speaker. And if you're having any internet or technical issues, please call in for sound at the number provided. It's also in the chat right now, or send a private message to GEMS, the GEMS participant in the chat. And so this meeting will be recorded, as I think you saw, and it will be posted for the GEMS members on our website. Okay. So we're going to go ahead and go to our first speaker. So our first speaker is Logan Everett. Dr. Everett has a BS in computer science from Binghamton University and a PhD in genomics and computational biology from the University of Pennsylvania. He joined EPA in 2019 as a bioinformatics scientist in the Center for Computational Toxicology and Exposure. His primary research focus is the application of high-throughput transcriptomics in chemical screening. Before working at the EPA, he was a senior bioinformatics scientist at SIOM, where he supported research at the National Toxicology Program on similar applications of transcriptomics for chemical risk assessments. He also has extensive postdoctoral research experience exploring multiple applications of transcriptomics and other next-generation sequencing assays. So Dr. Everett, we'll turn it over to you. Thank you for joining us.
Can you hear me? Yeah, okay. The mute control has gone away on my screen, but as long as you can hear me, we're good.
We can hear you.
Okay, great. And my slides are showing full screen?
They look good.
Okay, great. Thanks for that introduction and for inviting me to present here today. So I'm going to talk a bit about our efforts at EPA, specifically at the Center for Computational Toxicology and Exposure, or CCTE, to integrate transcriptomics in high throughput into our efforts to do chemical safety screening. So before I get into the actual research, I first have to give a quick disclaimer that the views expressed in this presentation are my own. They don't necessarily reflect the views or policies of EPA, and any company or product names do not constitute endorsement by EPA. And I also want to acknowledge before I start that what I'm presenting is the joint effort of many scientists at EPA. We pride ourselves on doing team science in CCTE, and the folks shown here have made critical contributions to the work that I'm going to talk about and continue to drive these projects forward on multiple fronts. So just to kind of set the larger context for this work, EPA is developing a tiered testing strategy for chemical safety assessment. And the first tier is intended to utilize assays such as high throughput transcriptomics that provide broad coverage for a wide range of potential hazards. But these assays need to be flexible and cost efficient enough to screen thousands of chemicals. And we would then use this data to prioritize chemicals for further screening and assessments in tiers 2 and 3. And in order to do this, we need to both estimate potency from the initial tier 1 data or identify points of departure for each chemical and predict the relevant biological targets to help guide those follow-up experiments. So identifying which orthogonal conformation assays to use, as well as identifying which adverse outcome pathway models to link into. And one other thing I want to note here is that this is kind of building on past work in our center where we were using something called the ToxCast suite of assays, which were a large suite of several hundred targeted assays that were high throughput. So these were both cell-free biochemical assays for chemical and enzyme interactions, as well as cell-based assays, but looking at a specific target. So we have a whole bunch of that data on some chemicals, but we're looking to accelerate things even further by basically being able to rely on just one or a few assays that give us really broad coverage and that we can kind of pull a lot of different biological endpoints out of and then focus the follow-up in tiers 2 and 3. So I'll first explain how we're able to actually scale transcriptomics up to be high throughput, and then I'll describe our current bioinformatics approach to address some of the unique challenges in handling data of this scale and complexity. So we're currently developing this framework for screening chemicals in human cell lines. So it's still in vitro chemical screening, similar to the ToxCast suite that has been used for a couple decades now. And in order to do any type of in vitro chemical screening rapidly and cost effectively, EPA has developed highly standardized and automated protocols for cell culture and chemical exposure. And this is on the lab side overseen by Josh Harrell, who's another scientist at CCTE. And so one of the key things here, there's a lot of different equipment that helps us automate this. In particular, we use acoustic dispenser technology, represented here by the lab site Echo, that allows us to expose cells in large plate formats to multiple concentrations of each chemical. And so it allows us to automate moving small amounts of liquid around on 384 well plates. And then we also, in addition to the transcriptomics, we always run some type of cell viability or cytotoxicity assay in parallel, which allows us to focus the analysis of the transcriptomics data on the concentrations of chemical exposure that are essentially pre-cytotoxic or sub-cytotoxic in their exposure. And again, the core assay for this is the targeted transcriptomics, which I'll talk about in a couple slides. But first, I want to just introduce kind of the general idea of our study design. So using that automated workflow that I just showed you, we can do rapid screening using 384 well plates. We use that automated liquid handling system to basically set up dose plates with 8-point dilution series of each test chemical. We can fit roughly a little over 40 chemicals per dose plate. And then that same liquid handling system is used to transfer each of those concentrations of each chemical onto a plate that is that has the cell line of interest growing on it. So we typically do one cell line at a time for each screening study that we do. And so this allows us to randomize all the positions of the different exposures, so we can break up any kind of confounding just plate positional effects. We also include multiple types of both reference treatments. So these are chemical exposures that are standardized throughout our studies and then still get transferred through the liquid handling and done as cell exposures the same way as our test chemicals, as well as QC standards that are basically bulk prepared samples that are added to each plate right before the transcriptomic profiling. And so these allow us to both look extensively at reproducibility in these studies, as well as identify problems at multiple steps of the workflow. And so each plate of test chemicals is run, the whole exposure process and transcriptomic profiling is run in triplicate. So we do essentially three plates of the same collection of chemicals and concentrations. But each one of those plates is randomized independently and run on a separate batch of the cell line to help with capturing variability in the cell line behavior. And so we've used this general experimental design for a pilot study, which came out just a few months ago in Toxicology. The reference is down here, but we've basically been using this same type of experimental design for larger screens of over 1,000 chemicals each in 3 different cell lines. We're currently analyzing and writing up all that data, and I'll give you some previews of what we have so far. But the point is that we're able to do this in an automated way at a pretty large scale. So our studies have dozens of plates of 384 wells. And one other thing to note here is that, just in the scope that I'm showing you here, right, for 40 plus chemicals, 3 of these plates is going to add up to close to 1,000 samples that we need to do transcriptional profiling on. So we really need our transcriptomics assay to be truly high throughput, right? A study where we're going to look at over 1,000 chemicals is going to generate tens of thousands of transcriptomic samples that we need to be able to process. And that's really not particularly easy or cheap to do with traditional RNA-seq. So in order to do this transcriptomic profiling at the throughput that we need to do, we're using an assay called TempoSeq that was developed by BioSpider. I'm happy in the Q&A, I'm happy to get more into the weeds of the chemistry of this assay and how it works. There's also a really good paper put out by the people at BioSpider explaining the assay in great detail. But the key thing to understand for what makes this assay different from traditional RNA-seq is that rather than sequence fragments from throughout our target RNA, we're just sequencing specific 50 nucleotide probe sequences that have been designed to match 50 base pairs in the target gene of interest. And so there are libraries of these probes already made that allow us to profile nearly all of the protein coding genes in the human transcriptome. And then the number of times that any given probe is sequenced gives us an estimate of the relative abundance of the target mRNA. And one key innovation to using this assay over traditional RNA-seq is that we don't have to purify RNA. You can use this assay, it is compatible with purified RNA, but it's also compatible with raw cell lysates. And that's critical for our large-scale automated workflow because we don't have a way to cost effectively do RNA purifications on those tens of thousands of samples. So we do this on the raw cell lysates. And there's the standard paired barcoding that we can do for Illumina sequencing that allows us to multiplex lots of samples into a single lane. And because we only need to sequence these 50 nucleotide probes instead of the entire fragments from the entire length of each transcript, we can use much lower depth per sample, which really cuts down the cost of the actual sequencing for when we do this. So we've now generated, as I was saying, data from 3 large screens, over 1,000 chemicals each in 3 different cell types. That's over 100,000 total TempoSeq samples. So we needed to establish standardized and efficient ways of processing all that data. And that overall pipeline is shown here. This is, again, largely been laid out in the pilot study that we already published a few months ago. And much of the source code for kind of pushing the data through the various tools is already up on GitHub. And so I'm not going to go through this step by step, but I want to just highlight a few critical steps in this pipeline and some key results that demonstrate the overall value of this approach. And I'll also point out here that we are largely relying on open source tools to do almost everything in here. So first off, one thing to highlight is that we've put a lot of effort into building comprehensive QC procedures into our analysis pipeline. So being able to skip the RNA purification step with the TempoSeq assay, as I said, is critical to being able to do this at the scale that we need to do it. But it also means that we're skipping some of the typical quality checks from traditional RNA-seq that we would have had done before even sequencing a sample, right? So in traditional RNA-seq, you typically look at input concentration or RNA concentration of your input material, as well as RNA integrity number. And that would allow you to filter out bad samples before you even go to the sequencer, or at least kind of have them flagged before you even start looking at the data. And so we don't have either of those working for us with the TempoSeq assay. So to address that, we've developed a battery of tests that we can use to flag and remove low quality TempoSeq samples based purely on features of the aligned reads and counts, and that those are represented by this blue box here. We also mask out samples, as I mentioned, where the chemical treatment was cytotoxic or failed, where it was cytotoxic, particularly where it kills more than half of the cells in a well. And then we also have ways to identify when the actual chemical dispensing doesn't work properly on the acoustic dispenser. So it has logs that identify those problems. And so all of these QC steps are summarized on the left here across the more than 100,000 samples that I mentioned. So it's split by the 3 cell lines that we've looked at. And you'll notice on the y-axis, we're looking at just a few percent here, right? So out of those over 100,000 samples, only a few percent give us any sort of problem in any of these categories, from the liquid handling of the chemical all the way to getting reliable data out of the TempoSeq assay. So the vast majority, even after all of this, all of these QC checks, the vast majority of the data, more than 95% of our samples, are usable for the analysis that we want to do. Another important piece to our pipeline is using statistical models that are designed for sequencing-based data. So it's been appreciated for many years in the RNA-seq field that the statistical tests that were originally developed for microarray data are not reliable, or not what you should be using for the count-based data that you get out of sequencing platforms. And so we're using DE-seq2, which is a really, at this point, very widely used biostatistics package in R that is designed specifically for count-based data coming out of sequencing experiments. So we use DE-seq2 to convert our raw probe count data into fold changes that are moderated to account for both differences in sequencing depth, or differences in sequencing depth across both samples and different expression levels of different genes. And the figure on the left is showing a global view of how many probes are differentially expressed per chemical according to some of the statistics that we can get out of DE-seq2 for the large screens in each of our 3 cell types. And so, and it's broken down by concentration that we've looked at, and basically each box plot is covering all chemicals, showing them by the number of differentially expressed probes at a 10% FDR. So on the left, you can see that for the vast majority of chemicals in any of our 3 cell lines, the lowest concentration that we are exposing the cells to really does not perturb any genes with any significance. These are just, there are outliers here. So there are some chemicals that are highly potent, but the vast majority are showing no activity at our lowest concentration. And then as we move up our dilution series, or our concentration ladder here, we see an increasing or accumulating number of differentially expressed genes or probes. And so at the highest, our standard highest concentration of 100 micromolar, roughly half or more than half of the chemicals in each cell type are giving us some amount of transcriptional perturbation, often on the order of hundreds to thousands of individual genes being perturbed. And so this overall trend is encouraging that we're seeing, we've basically hit a concentration range for most of these chemicals where we go from almost no transcriptional perturbation to lots of statistically significant transcriptional perturbation with, increasing amounts in between. So that's kind of the 30,000-foot bird's eye view of the data. But what we really want to do is drill down and look at per chemical, what is the concentration responsive behavior? And in particular, we are interested in finding expression changes that are coordinated across biologically relevant gene sets, because those we can actually make some amount of mechanistic interpretation out of, and for which we can fit standard concentration response curves that allow us to estimate a point of departure. And so to achieve those aims, we first take the moderated fold changes on the log2 scale that come out of DE-seq2. We feed those into a gene set scoring method, such as initially this was done using single sample GSEA, although we've also found that a much more simplified, just taking a difference of the average fold changes in and out of a gene set works just as well for our purposes. But either way, we take those scores, we compute them for a collection of signatures that we've put together that focus on, that are curated out of several public sources, but that focus on biological processes and molecular targets with toxicological relevance. And this catalog is maintained and curated by Richard Judson and Laura Taylor at our center. And so one observation about these signature scores is that they are more reproducible than the individual fold changes, right? So there's some amount of noise in these fold changes. We actually have, so this is looking at 3 reference chemicals that were run at single concentration across every single plate in our large screen of chemicals in MCF7 cells. So we can basically take every set of triplicate plates and do DE-seq2 analysis on each of these 3 chemical treatments. And that gives us essentially 37 independent replicates of analyzing, or independent experiments of analyzing the response to each of these 3 chemicals. And then we can look at how well correlated the fold changes or the signature scores are across all of those replicate experiments. And what we find is that particularly for treatments that elicit a weaker overall transcriptional response, such as the particular concentration of genistein that we used here, the correlation of fold changes is positive, and it's basically moderate correlation, but that correlation gets much better when we convert these individual gene fold changes to signature scores. So these signature scores are helping us clean up some of the noise, get better, more reproducible signal. And this is particularly important the weaker the overall effect size of the treatment is. So it's these signature scores that we think are more useful to actually do the concentration response modeling on. And so to do that, we take those individual signature scores at each concentration of a test chemical treatment and feed these into the latest version of a curve fitting package called TCPL. And that results in signature level POD estimates. So once we have a curve fit and it passes kind of some basic statistics to tell us that there's activity, we can identify things like the benchmark dose as the point of departure for that particular signature in that cell type in response to that chemical. And this is, again, methodology that's being developed by Richard Judson at EPA. And so so this gives us sort of still a really large amount of data, many different signatures across many different chemicals, and we're still working on how to best summarize that for our larger screens. But in our pilot analysis, just using MCF7 cells, we found high concordance with previous results from the earlier ToxCast studies that I mentioned at the beginning. So I'll just spend a couple minutes just explaining this figure, and then I'll wrap up for questions. So in a recent paper from Katie Paul Friedman at our center, she developed a method, this work developed a method for kind of taking all of that earlier ToxCast data. So again, these are many different targeted high throughput assays, and getting an overall POD estimate for a chemical, basically taking the fifth percentile of the various POD estimates that come out of all these different assays. And so that's what's for the chemicals in our pilot analysis. That's what's captured on the x-axis here. And each of these dots is one of the chemicals in our pilot analysis. And so we basically did a very similar process with our signature level PODs that came out of the high throughput transcriptomics data to get an overall chemical POD. And for the majority of chemicals in our pilot analysis, the concordance was quite high. So within tenfold in either direction between the ToxCast POD and the transcriptomics POD. And this is again using the signature level PODs. And for many of the chemicals that did not match up as well, many of these can be explained by the fact that the known target for those chemicals is missing in MCF7 cells. So we might expect that if we screen them in a different cell line, we should actually be able to pick up something that's concordant with the original ToxCast data. But overall, this is encouraging that it suggests that we can use transcriptomics as potentially a replacement for the much more expensive and time-consuming suite of ToxCast assays that were run previously. So just to wrap up, at EPA, we've developed a reliable and cost-efficient workflow for generating high-throughput transcriptomics data from thousands of chemicals across multiple cell lines. As I just mentioned, we've gotten results that are overall concordant with previous assays and efforts for doing high-throughput screening. And then the upcoming research really is going in a whole bunch of directions at once. So I mentioned we're still analyzing and writing up the data from our larger screens. And even while that's going on, there's more data coming in. So we're still continuing to screen in additional cell lines and continuing to explore additional methods that might be used to get more informative results out of this type of data. So better methods to summarize signature level and overall PODs from this type of high dimensional data, as well as models to predict the molecular initiating event or the specific target relevant to toxicity for each chemical. And so I'll just acknowledge again, this work is a large team effort. In addition to the team members that I showed at the beginning, I want to thank additional collaborators at EPA and our center leadership. And thank you all for your attention. And I'm happy to take questions now. And I also have my email here for anything we don't have time to get to today at the GEMS meeting. So thank you.
Great. Thank you for your presentation. Does anyone have any questions? Again, you can raise your hand or type them in the chat. I can get started off with one initially. Wondering, I guess sort of a logistical question. How do you have to send your samples off to Biospider, for instance, or can you run that in-house?
So we do send them to Biospider.
Okay.
And that's partly because we don't have a lot of Illumina sequencing in-house, but I believe they sell kits that you can do it yourself, but then you have to figure out how to do you either need to have the Illumina set up in-house or you're going to end up sending off your samples to a sequencing core. But yeah, I believe, yeah.
Okay, sorry. So I guess they run both the whole transcriptome assay and then they sequence it for you as well and send it back. Is that correct?
Yes.
Okay. But are the rest of the portions run in-house, I take it? So you send the lights and then they perform the assays for you.
Right,
okay.
right. So all of the cell culturing, the automated chemical exposures, that's all done in Josh Harrell's lab at EPA.
Okay, great.
Yeah.
Thank you. All right, looks like Caitlin, your question?
Yeah, hi. Thank you, Dr. Everett, for the presentation. I was just wondering, how did you guys initially vet and determine which chemicals that you would use for these assays?
So there is a lot more history to that goes back before I even came to EPA. So there's something called the ToxCast library, which was originally put together to basically build a library of chemicals that would be useful for testing and validating any new high throughput screening assay. And again, this goes back to the original ToxCast suite, which was kind of this growing catalog of targeted high throughput assays so for the most part, the chemicals that we've tested so far are coming from that library. So a lot of them are, it's kind of been selected to cover a wide range of different types of compounds. There's pharmaceuticals in there with known targets. There are environmental chemicals that have known hazards. And then we are increasingly including chemicals that have new concern, that are sort of recently on the list of concerning chemicals. So we have some parts of the more recent screens that we've done that include roughly 150 PFAS chemicals specifically that we've gotten to, that we've brought into the lab that we're able to handle well enough to do these kinds of cell exposures. But it is a constant, I mean, it's a constantly changing or moving target of sort of which chemicals to prioritize to put in these screens in the first place. And that there's a lot of, I think, politics that goes into that as well, is the short answer.
Fair enough. Thank you.
Yeah.
Okay, I think next is George.
Yes, thank you. Very interesting presentation. I've seen a lot of it before and have to admit that I'm not a transcriptomic expert, but I have a question related to this. Do all transcription signals have the same weight? In other words, do some transcription products have a stronger biological effect than others? For example, hormones, which are pretty potent, biological compounds. So at any rate, just to, can you comment on that?
Right.
So kind of get that.
Yeah. So theoretically, yes. Right. Some transcripts are going to be much more important than others. We don't have any systematic way to really build that in, because the reality is our knowledge of that is very, very far from complete. But so there's a couple things that factor in. One is that the way that the DESeq2 statistical models are being used, there's definitely going to be more of an emphasis in the end results on genes that are giving us essentially good signal to noise. So the noisier that a particular gene expression signal is compared to the amount of response that we're seeing in response to the chemical treatment, the noisier that is, the less, it's basically going to kind of squeeze those fold changes to zero, and we're just kind of not going to look at those genes because they're not responding in a way that's actually robust to the noise. And that, that noise could be completely orthogonal to biological relevance, unfortunately, right? It could have to do with just the design of a particular probe in the TempoSeq assay. And then the other thing is that because we're using these catalogs of gene sets that we know are, have been curated for toxicological relevance, genes that something that is, say, a hormone that is known to be really intrinsic to some important process that responds to toxicological stress, that's going to show up in more gene sets, and so it's going to be more likely to actually contribute to a signature that we're actually going to see a good curve response and a potent response for that particular signature. So in that sense, genes that are showing up in more of the existing gene set catalog are going to get more weight in the final results, just because they have a higher chance of hitting somewhere, or that signature actually being the driver for picking our POD. But we don't actively weight genes based on any kind of biological importance at this time, because it's really, really challenging to do that in a consistent quantitative way for the whole transcriptome.
Right. I'm just wondering if eventually, looking down the road, if there becomes a method for doing such, it might help to tighten your correlations too, because you would then, be looking at those things that are probably the most relevant for a biological response, and not just what's being, expressed more in the transcriptome. But that's just, pie in the sky, down the road, a thought experiment at this point.
So down the road, I think what we hope to do is figure out which signatures are actually predictive of hazards that we, that we can actually link to hazards of concern, right? Because there may be transcriptional changes that, are a coordinated set of genes that we can link to some biological process, but it doesn't actually mean that the cells can't recover from it or that it's doing anything bad to the cells. It's just how the cells respond to process that chemical back out. So I think that's where we see that going, is building up some evidence of which signatures are actually predictive of a hazard going forward.
Okay, well, thanks for understanding my question.
Yeah.
Okay, Ramesh?
Thank you. So very nice presentation. So I think you touched my question partly towards the end of the last answer. So in terms of the prioritizing versus predictive, at the transcription level, there's still a debate in biology how much it is translated to protein, whether it is just perturbance in a cell-specific manner and an approach-specific, meaning in vitro versus in vivo, to gain that confidence or the robustness of the data to use this as, say, for a predictive. What additional endpoints you think it would add more value? And is there any effort made at EPA or elsewhere to include either proteomics or in vivo correlate of a selected compound with a response in the transcriptomic with a reasonable POD to validate those assays at biology?
Right. So yeah, that's a great question. Thank you. There's no effort that I'm aware of to do proteomics anywhere near the scale that we're doing transcriptomics, because it just can't be done in a remotely cost-effective way, right? With infinite money, it would be wonderful to look at that. But so one way to think about it is that any transcriptional changes we're seeing, particularly transcriptional changes that are coordinated across some set of genes, are reflective of something upstream that is changing, right? So some signaling pathway that ultimately impacts transcription, maybe perturbation of a particular nuclear receptor. So it's actually less about what are the how are these transcriptional changes turning into proteins, turning into effects, and more about the, if we see a whole bunch of estrogen receptor targets change their transcript levels, then that probably means that the chemical hit the estrogen receptor or some part of that signaling pathway. But in terms of actually relating it to something some downstream effect, probably the area where we have the best potential to do that is that we're also using that whole automated screening setup that I showed you at the beginning to do cell painting assays, where you can actually look at basically high content imaging of the cells with several different dyes that give you fluorescent signal for the nucleus, the Golgi bodies, the mitochondria, and we have a whole other data set where we can look at that. So it ends up being over 1,000 different image-based features of the cell phenotype in response to some of these same chemicals and concentrations. So that's an area where we could look at, which transcriptional changes actually mean there is a change in cell morphology happening, which would, several steps removed from the transcriptional change, but it's some kind of phenotypic, downstream effect of transcription.
Thank you.
Yeah.
Do you have any other questions? I have one additional one. So apologies if you mentioned this before, but how did you pick your 3 cell lines?
I did not mention that, and I don't even think I put a slide in to address that. But so MCF7 was chosen just because it's really easy to work with in the lab, and it's often used, and it does express a whole wide array of different nuclear receptors and endocrine receptors. U2OS is in there because it was the initial cell line that was used for the cell painting assay. That happens to grow really well in monolayer cultures, which are really easy for imaging. So we wanted to have corresponding transcriptomic data for that cell line. And then HEPA-RG was included because of the relevance to liver toxicology.
Yeah.
And it is a metabolically competent cell line, unlike HepG2 for the most part. So, but in terms of going forward, how do we pick additional cell lines? We have actually done just baseline transcriptomic profiling on something like 40 or 60 different cell lines. And there's a paper that that Josh Harrell is in the process of putting out now, where they've done analysis to, they've created a method to basically say, okay, which cell line is going to give us the most new biology compared to the 3 that we've already profiled, right? Or at least is predicted to give us the most new biology just based on how different the expression patterns are in that cell line. So that's how we're picking future cell lines going forward.
Yeah, makes sense. Great, thank you. Okay, I think we have time for one more question. Errol? I think you're on mute. Have your hand up for a question if you have one.
Okay. I think you may have answered some of my question, but I'm concerned about the fact that the HEPA-RG, as you said, is metabolically competent, but the other 2 cell lines are less so, if at all. And do you see what is the point of using cell lines that are not capable of metabolism of these sample chemicals, or did you pick chemicals that do not require metabolism?
So many of the chemicals in our screens do not require metabolism. I mean, we're seeing transcriptional.
Was that deliberate?
I don't know if it was deliberate in terms of the selection of the original. I mean, I think a lot of the things that are in the ToxCast library, that was deliberate, because at the time that library was first put together, there were not a lot of assays in metabolically competent cell lines. I mean, that's a fairly recent to be able to do that at all. And there are other labs at EPA that are working on basically taking any of these cell lines and doing this assay in a way where there's also microsomes printed on basically a little spike that comes down from the cap of the, basically the cap that you put over the well, that would kind of add metabolic competence to any cell line that we want to do the testing in. But right now, yeah, it's a limitation. But what I can say is that we are seeing transcriptional responses to over half the chemicals in the screen. So there's a lot there that doesn't require any, it doesn't require being metabolized to have an effect.
Well, it could have an effect, but a different effect if it's not metabolized.
Yep. Yeah, that's true. Yep, that's true.
Okay, Amy, I think we have time for one more quick question, please.
Yes, actually, this is a comment on the previous question. I thought by having cells not being able to metabolize the chemical is actually not too far away from the in vivo condition. Not all of the cells generally metabolize by themselves. With the circulation, they receive metabolites from other tissues. So as Logan just mentioned, having additional component outside the cell type of interest to generate metabolites might be a good mimic system.
Right, we would just need to then, if there's something where it's really a metabolite that is has more toxicological concern, we would then need to have that purified metabolite and do the testing on that. And that's available in some cases, but not always, is I guess the short answer to that. But yeah, it's a good point, right? Cells in other parts of the body are not going to be metabolizing these things themselves. They're either going to get exposed to a metabolite that came out of the liver or the chemical itself. So.
Great, thank you.
Yep.
Thanks very much, Dr. Everett, for your presentation.
Thank you.
Appreciate it. Okay, our next speaker is Leah Wehmas. So Dr. Wehmas has a BS in cell and molecular biology from the University of Minnesota, and a PhD in environmental and molecular toxicology from Oregon State University. She joined EPA Center for Computational Toxicology and Exposure as a genomics scientist in 2019. From 2016 to 2019, she was a postdoctoral fellow at the EPA, where her research focused on understanding modes of action of chemical carcinogens using FFPE samples from archival studies. Her current research focuses on toxicogenomic approaches to help characterize chemical hazard, with an emphasis on new approach methodologies. So this includes repurposing archival research samples for next generation sequencing, using cell-based assays and new gene editing techniques to better understand toxicity, and also quantifying biological potency through genomic dose response data to advance traditional chemical risk assessments. So I will turn it over to you now. Thank you very much, Dr. Wehmas.
Yeah, sorry. I'm just trying to share my screen properly here. I thought it was working, but okay, let's see if this, I'm trying to get it into full screen mode. Not sure why it's not. There we go. Okay,
Yep,
it's just
that
being
looks
slow.
good. Thanks.
Okay, thank you so much, Katie, for that introduction. And I'd like to start out by thanking GEMS for inviting me to speak today on a case study using transcriptomics from acute in vivo and in vitro exposures to inform points of departure. Before I get into the remainder of my talk, I would like to acknowledge contributors up front, especially Nissa Tucker, as well as Jermaine Ford and the Chemical Safety for Sustainability, which supports this research here at EPA. But as a government worker, I have a disclaimer that the views presented in this talk are mine and don't represent the agency. So what are points of departure? I know Logan gave a great talk about a lot of the high throughput transcriptomics we're doing here at EPA. I'm going to focus in a little bit more on one, I guess you'd say later in the tiered testing strategy, but how we're relating in vivo and vitro points of departure to understand thresholds of chemical toxicity. And we use points of departure at EPA to set the threshold for chemical toxicity. And the point of departure is used, or it's derived from the dose response assessment, which happens after hazard identification of a particular chemical. That's compared to possible exposure, and we use that to characterize risk for chemicals. The dose response assessment can be done a couple ways. There's the no observed adverse effect level or the lowest observed adverse effect level approach, but this isn't the preferred method for establishing risk thresholds, as it depends on the dose selected in any one particular study. We much more prefer to do benchmark dose modeling, which is much more quantitative. And from the benchmark dose, which is just the dose level that causes a significant change in response at a 10% response rate above control, that's used with the lower confidence limit to set the point of departure. And so we use the lower confidence limit of that benchmark dose because it's health protective. Okay, my slides, trying to advance. Ah, there we go. But why are we, or how do we use these points of departure in the AOP framework? Well, and using new approach methods. So I'm interested in using new approach methods and the AOP framework to understand toxicity, with the increasing focus on these higher throughput assays like the ones Logan described. There is a need to link that new information with more traditional endpoints of concern at, say, the individual or even population level. We can use existing archival studies, which is something that I work on, or just existing data sets from animal studies and transcriptomic information to help bridge that gap across the different levels of biological organization. And then we can also move into understanding how transcriptomics and concentration response assessment in vitro can be used to predict risk or assess risk of chemicals and how that translates to adverse effects in humans. But there are definitely some uncertainties in using transcriptomics to in vitro and in vivo to characterize chemical risk, but specifically for in vitro assessments, and this is just a figure taken from our next generation CompTox blueprint that came out in 2019, that in order to gain regulatory acceptance of using some of these new approach methods and transcriptomics-based methods in vitro, we need to better understand uncertainties in chemical delivery. Also, as we just discussed with the previous talk, metabolism may be an important factor, as well as then just understanding differences based off of the different assays or platforms culturing in a monolayer versus a whole intact organism. And so I'm hoping, or what I wanted to do with this case study is to use the existing acute in vivo transcriptomics to compare to the in vitro gene response to better understand adverse outcomes and predict risks, environmental health and safety risks. And so to do that, this is just an overview of the original in vivo experiment, as well as the secondary in vitro experiment that was done for comparison. And we're fortunate enough to have some existing transcriptomic data from a very well run study by April Lake, in which male mice were exposed to multiple dose levels of di2-ethylhexyl phthalate in the diet for 7 days, and they underwent total RNA sequencing. And I was able to then just reanalyze this data with an updated annotation model for comparison to my newly acquired transcriptomic data from the in vitro portion. And so with the in vitro part of the experiment, I used strain matched primary liver cells, hoping to maintain at least some of the liver metabolic capacity in this assay, and then expose those cells to multiple concentrations of DHP, 5 different concentrations. And I also included MEHP in this exposure because MEHP, or mono-2-ethylhexyl phthalate, is one of the major primary metabolites of DHP in both humans and mice. And however, it's believed that most of the metabolism actually occurs in the gut. So by including that as a control in this particular assay, we could also look at whether gut-based metabolism versus liver metabolism was important in the transcriptomic response. And then to help assess dosimetry with these chemicals, we collected the cell medium and froze that down for analytical chemistry. And Logan gave a great overview of the tempo sequencing assay, and that's what I used for these primary liver cell exposures. It's very convenient to be able to then lyse the cells after collecting the media and then doing this targeted RNA sequencing approach. And so let's see here. This was just all to then, it's very, I guess, exploratory analysis. We got this done right before COVID hit. So Some of the analytical chemistry results aren't in. They're actually just getting to looking at the media for me, so I won't be able to discuss that. But I will discuss the transcriptomic results using the nominal concentrations. So what we saw in vivo is something that we've seen previously in published studies in Lake et al. But that exposure to multiple dose levels of DHP resulted in a nice increase in significant gene expression. And I'll just give you an overview of this particular plot. It's called an upset plot if you haven't seen it before. It's a nice way to show a lot of intersections in genes as opposed to a Venn diagram. And so on the left-hand side, I have the genes separated by those that were upregulated at each dose level, and then those are colored yellow, and then blue is the downregulated genes. And just if you look at general significant differentially expressed genes, there's a nice increase, especially amongst the upregulated. And then on the right-hand side of the plot, the solid connected dots are indicating where there's an intersection. And the bar above that indicates the number of genes that are shared. And so if you go to the far right, you can see that all of the, across all the dose levels, there were 148 significantly upregulated genes that were shared amongst those groups. So that's a very good, robust response. And then if you just look at some genes known to be involved in DHP's mode of action, which is peroxisome proliferation receptor alpha activation, we see that these acyl-CoA thioesterases are significantly upregulated, several of them. And for ACOTS 1 and 2, it's a nice dose trend that's apparent, which was all seen in the previous study. And then when we take those significant genes and actually map them to the gene sets, as Logan described in a very nice way in his talk, and we look at the lowest median gene set benchmark dose from our DHP study, we see that it's 1.4-fold lower than the BMD set for the 2-year hepatocellular adenoma or carcinoma. And so what this is showing, and what they previously showed in Lake et al., is that this is highly consistent so a 7-day exposure results in a transcriptomic benchmark dose that is very consistent with the results from a 2-year assay measuring more traditional endpoints like carcinoma or adenoma. And if we look at the GO description, or the lowest median GO process, it's actually acyl-CoA metabolic process, which is nice to see with some of this work. It doesn't necessarily mean that the lowest median one is going to actually fit with your chemical's mode of action, but it's a nice thing to see. So for the in vitro study, what we saw is that the response wasn't quite as robust as what we saw with the in vivo experiment. I would say that that was not necessarily unexpected, but we do see that there are several upregulated and downregulated genes from the DEHP exposure. But if you look at the ones that are shared across groups, there aren't quite as many as what we saw in vivo. So there's maybe one or a handful of genes that are shared across some of these different exposure concentrations. And for MEHP, we're seeing a little bit more of a perhaps dose trend with just the total significant differentially expressed genes. As you increase concentration, we're seeing more of an increase in upregulated and even perhaps downregulated genes. But there's still not a whole lot of genes that are shared across all of the different exposure concentrations, but they're still there. And of course, this is just showing across both of the groups if there was any common genes, which is once again, like just looking at the highest 3 concentrations. When we look at the acyl-CoA thioesterases, what we see for DHP is that we actually didn't see any of those identified in the data set as significantly differentially expressed. However, we do see it with the MEHP. In fact, it's at the highest exposure concentration, we see a level of induction of ACOT1 that was similar to the lowest dose level tested in the in vivo DEHP study, which is suggesting that indeed gut-based metabolism could be an important factor in contributing to this response and might be something to consider when doing some of these cell-based assays, in addition to using metabolically competent liver cells. And then for ACOT2, we didn't see, or its level of induction wasn't quite as similar as the lowest dose of DHP. But when we take those significant differentially expressed genes and then map them to the pathways using benchmark concentration modeling, we identify the lowest median benchmark concentration for DHP of about 0.5 and micromolar and 11.7 micromolar for MEHP. And I am still in the process of converting these concentration responses to plasma-based dose levels using reverse dosimetry. That's been actually made a bit challenging for this particular chemical because di2-ethyl hexyl phthalate is a common component in plastics. And so use of a lot of these plastics in some of these in vitro assays and the cell culture systems makes it a little bit harder to do some of these measurements, but that is ongoing work. And so when we compare across all of our experiments, in vitro and in vivo, we see that there is a modest overlap of genes between the DHP in vitro, MEHP in vitro, and DHP in vivo. But there's definitely a more of a either assay or platform dependent overlap between the two in vitro experiments with 208 shared genes. So it's interesting, and I'd like to look a little bit more at some of these shared genes. They're definitely genes known to be identified in other DEHP exposure experiments, but I would like to look more closely at if their response induction or reduction is consistent with the in vivo portion of the experiment in a little bit more detail. So in conclusion, I'd say the cell culture response was perhaps a little less robust than our in vivo study. And there was definitely some assay dependent effects that are apparent at the transcriptomic level, but as we've seen, in vitro gene expression can be used to set points of departure and establish some idea of a risk threshold, as Logan and Josh are doing in a very high throughput manner. But there are some other factors that may be useful to consider with some of these exposure situations. And I'm hoping that these types of studies will help us better design, or hopefully maybe some of the lessons learned from this case study and others like it to help Josh and Logan with their efforts at doing some of these high throughput assays to better design it so we can get our in vitro transcriptomic points of departure and the genes and response levels more similar to what we would expect in vivo for human health and safety assessments. But I am definitely looking forward to working on getting the reverse dosimetry results so that we can make a better comparison of these points of departure across the in vitro to the in vivo dose thresholds. And of course, the one other conclusion, at least that I've learned from this, is that of course heavy use of plastics can create added challenges. I did include a lot of blanks and different types of controls on my plates to help with this, but there's so much background level DEHP, I think, from some of these heavy use of plastics that it's going to make the analytical chemistry a little tougher than we originally thought. And so with that, I hope I've shown you how we're using existing case study data sets and transcriptomics in short, acute in vivo exposures, as well as in vitro cell-based assays to better link thresholds of toxicity earlier, faster, maybe more efficiently to better help us assess the risk of chemicals, and then hopefully then translate that to human health and environmental risk and safety assessment. And so with that, I'll take any questions that people have.
Great, thank you so much for a really nice talk. Does anyone have any questions? I can start us off with one. I'm wondering how you are, how do you think you might be able to get around that plastics problem? Is there an option? That's pretty much what's in the plates, right? And
yeah, we'll see. I'm hoping that a lot of my controls will help with that, but I haven't heard back from the analytical chemist. I know that I had asked our in-house toxicokinetics expert, Barbara Wetmore, about the plasma-based concentrations for reverse dosimetry, and she said that it's just a bear to do DEHP. So I'm going to look more through the literature to see if somebody else has had success with it. But I don't know if it's a challenge. I mean, there are the option of glass plates, but that's not going to be cost effective. So I'm hoping that my blanks helped control for some of that.
yeah, absolutely. I have another question. So I was wondering about sort of like your exposure time. So you're looking at the correlations, for instance, a 7-day in vivo study, which presumably has like that chemicals at steady state, et cetera. So then how did you pick sort of the 1-day in vitro? Is it some of it sort of like logistical considerations and yes?
Yeah, it was for, well, logistical in that the primary cells that I used maintain their metabolic capacity. It goes sharply down after like, I think 48 hours, first of all. And so we wanted to make sure that the cells were still maintaining their liver metabolism, just in case if that potentially influenced the results. And then a lot of the in vitro data sets are just run for 1 day. And so initially I thought about doing a repeat exposure, but I was like, I just don't think that that's going to make these results. If we want to try to inform, like say some of the higher throughput or provide advice for some of those experiments and design, it would be better to have it be consistent with some of their methods. And so that's kind of how it was ease of use and limitations of the cells as well. So.
Great, thank you. Okay, Errol, please go ahead.
Okay, this was a nice set of experiments. I appreciate it, but there may be plastics that do not have phthalates in them, and that would be one way to go. But coming back to the basics, and you may have already addressed this, these approaches are designed for unknown chemicals, without chemicals whose toxicology is not yet known. Well, when you compare the in vitro and in vivo patterns that you've gotten, would you know without the names that they were the same chemical or the same class of chemical? You may have already addressed this, but.
So I'm not totally sure if I understand the question. No, so the point of this experiment was to look at known toxicants.
I realize that you use known toxicants to set up the system.
Yeah.
And to prove the system. but are the results you got from the in vivo consistent with the results you got from the in vitro? In
So,
other words,
no,
is the in vitro predicting the in vivo response? And to what
so that's okay. Sorry. Yeah, I think I understand now. Sorry. That's a good question. So it's still a little bit up in the air because I'd like to do the reverse dosimetry so we can compare at least our benchmark doses on a mg per kg per day dose level in the in vitro and the in vivo. But at least for the gene response, I would say that there seemed to be more of a either platform or assay effect, and the actual known DEHP effect wasn't so robust in this. And that may once again be partly because of the assay. It's a monolayer cell culture, but also there's the factor of the metabolism, the gut-based metabolism. But that's why I would like to compare to see if the points of departure and the benchmark doses are actually consistent with each other. But when you just look at the gene levels, the overlap isn't super robust.
Yeah.
So the in vitro is not necessarily, if I didn't know anything about the in vivo data or didn't know anything about DEHP and went off of the in vitro alone, I feel like some of those genes that are known to be involved in DEHP's mode of action may have gotten, I don't know what you would say, like drowned out in the noise of the platform effect. What I need to look a little bit more closely is how much were those shared genes induced. If it was like a very robust level of induction in those genes, then maybe those would show up as being like, okay, these are key drivers in the concentration response. So this is still definitely ongoing in data analysis and interpretation.
you might want to consider something that has a more simple in vivo metabolism profile, such as dimethyl or diethyl nitrosamine, which we know is primarily activated in the liver. So you don't have any competing in vivo processes that you don't have in vitro. You're not worried about gut metabolism or other cell metabolism. So
yeah, definitely a good suggestion. Thank you.
Okay, great. We don't have any other questions. I want to thank you very much for your presentation, Dr. Wehmas, and any other questions that come up, please feel free to add them in the chat. Right now, we're going to go ahead and take a break until 2:25. So please come back then, and we'll then Tina Stevens will give us her last transcriptomics talk. Thank you. Speaker is great, thank you. Our next speaker is Tina Stevens. Dr. Stevens earned her PhD in toxicology from the University of North Carolina at Chapel Hill, and she's a board certified toxicologist. She currently works at Syngenta as a technical expert in the Human Safety Risk Assessment Group, where she supports a portfolio of active ingredients that are currently registered in North America. She serves as the benchmark dose and residential risk assessment expert in that group. She also leads the BMD Express effort within Human Safety's Toxicogenomics group. She has prior experience working at EPA's National Center for Environmental Assessment, where she conducted human health risk assessments to set national ambient air quality standards. She also worked at Symetrica, where she was a senior toxicologist managing the FDA portfolio research on e-cigarettes. She was also a postdoctoral fellow at EPA studying the cardiopulmonary health effects of air pollutants. Thank you very much for joining us, and I will turn it over to you.
Thanks, Katie. And thank you, Gems, for inviting me to speak today. I'm going to turn off my video just in case. I've been having some issues with internet. So just to make sure things go smoothly, I'm going to turn off my camera. Okay, so today I'm going to talk to everybody about a case study of using transcriptomics in an ag chem case study. See if I can see why I'm not advancing my screen. There we go. So to give you a little bit of background about ag chem products, it takes a very long time and a lot of money to get a product to go from discovery into sales. So it can take around 12 years and cost somewhere in the range of $280 million. And I just want to focus here on this, on the toxicology piece on this diagram here, which shows it takes about 8 years to get a compound ready for market. And we can start off with somewhere around 150,000 compounds, which is quite a lot, all the way down to one, which would be our candidate compound. And it will take quite a bit of money as well as time and animals in order to complete that journey. And so we're always looking for ways of, while not compromising safety, but looking for ways to get a product to market quicker. And so normally the way we do studies, we've changed them up a little bit, and we're now doing a little bit more robust studies in this early phase, in this development phase, and having a really robust studies. So doing toxicogenomics as well as the standard toxicokinetics and clinical observations and pathology and hematology and clinical chemistry. And we're doing this in order to, this will allow us to integrate the systemic exposure and effects and enabling hypothesis generation to streamline subsequent testing and allow for more quantitative benchmark dose analysis so that we can predict dose settings and estimate our chronic point of departures, and also to reduce the generation of irrelevant high dose hazards. So I'm going to walk you through a case example. I'm going to call the compound X for this presentation. And so this is the in-life study that we conducted. So we did a 14-day dietary study in rats, and we looked at several different doses. So we had 0, 100, 300, 1,000, 3,000, 4,500, and 7,000 ppm. And this was, we were given them in a dietary study, and there's 5 per sex per group. And we looked at all the other different parameters that I was telling you about. And for the toxicogenomic piece, we were looking at we're doing FFPE samples and using TempoSeq technology that was talked about earlier today in order to do our transcriptomic analysis. So we weren't, for this, we were using our, we're using histology blocks, and we were not isolating RNA. And the goal of this study, again, was this hypothesis generation. So we were looking to see, to do regular transcriptomics and look to see if, what are the potential target organs, what are functional pathways associated with this exposure, and are there any mode of actions that can be identified or excluded? And then in addition, we're also wanting to use BMD Express to inform the dose settings for the 90-day study and also for early stage risk assessments. And so for the results of the in-life study, we saw no treatment-related deaths, no clinical observations, no effects on body weight, no effects on food consumption. And we did show that the TK results were showing dose proportionality. And there were no test item related microscopic findings. There were, however, increases in liver weight. And if you can see these graphs on the right, you can see that there were increase in liver weights. This is for the male, and then this one's the female. We did see increase in liver weights, and benchmark dose modeling was performed on this, and the BMDLs were 18.45 and 43.83. And so for the transcriptomics, like I said before, we were using the TempoSeq technology. And so the first step was to do QC. And so in this step, we had some issues with the forced stomach. So in this study here, we took a number of different samples of tissues. So we had adrenal, bone marrow, forced stomach, heart, kidney, liver, lung, pancreas, spleen, testes, thymus, thyroid, ovary, and uterus. And we had 5 sex per group, but not all of them were considered quality samples. And so these were the ones that ended up coming through for analysis. And so just to give a quick background on the genomic benchmark dose conceptual model. So all apical effects result from a prior change at the molecular level. So the generic adverse outcome pathway is that you have exposure, you have a molecular change, and then you have this apical effect. And with genomic benchmark dose modeling, the changes can be detected earlier. And so you're able to predict early on some chronic effects. And so this is the study design for the benchmark dose express analysis. So I had the rat and then multiple tissues, and using TempoSeq, we did the QC and normalization step. We visualized the data and were able to discard additional samples. We had a pre-filtering step. For this step, I used the Williams trend test. For the fitting dose response models, this is where I set the BMR and other parameters. And here, for this example, I set the BMR to 1. And then mapped the genes. And for this one here, I mapped them to the GO pathway analysis. And then I identified genes active as having greater than or equal than 3 genes and greater than or equal 5% of the pathway. And that would be considering a gene set active. And then I would report that number, and that number would be called the BPOD, or the biological effect point of departure. And so after going through this step, the only samples that passed in the male were the bone marrow and the liver, and then in the female were the adrenal gland, liver, ovary, and spleen. And so then this depicts the transcriptomic point of departure results. So here, you can see here I put in the liver weights. These were the apical effects. And I only had the BMDLs, so that's all that's reported right here. And then you can see below that are all the transcriptomic point of departures. And so here you can see that the point of departures were more sensitive than the apical effect seen at the 14-day, and that the bone marrow was the most sensitive in the male, and that the adrenal was the most sensitive in the female. And then we went on to go to do pathway analysis. And so the only samples that passed that had any dose responsive genes were in the liver. And what we saw was that it looked like it was a CAR PXR. Pathway was, there was pathway signaling. And then also it looked like some fatty acid metabolism. And so, so in summary, so the transcriptomic point of departures were identified for liver and bone marrow in the males, adrenal, liver, spleen, and ovary in the females. We saw modulated genes were observed in several organs, but only a dose-responsive genes were observed in the liver, and that these pathways were associated with CAR PXR signaling and fatty acid metabolism, and then also some oxidative stress response. Given this, we looked at the liver B pods and used those, which were 15 mg per kg per body weight per day in the males and 27 mg per kg body weight per day in the females. And since these were consistent with the CAR PXR findings as well as the liver weight changes, we use those to set the dose levels for the 90-day study. And we also use them to predict the chronic endpoints in our early stage risk assessments. So this brings me to the 90-day dietary study design. So here, again, we used male and female rats. This time we used 10 per sex per group. And we had our lowest group was set by the transcriptomic point of departures. So they were given 0, 225, 750, 2250, and 4500 ppm. And so for the endpoints here, we had very similar to the endpoints before, except this time we're looking at metabolomics rather than transcriptomics. So again, no mortality, no abnormal clinical signs, no effects on body weight or food consumption. The TK results were generally dose responsive, were generally dose proportional. There were no exposure-related hematology findings. There were significant clinical chemistry changes in LDL and globulin at 2250 and 4500. And there was significantly lower T4 levels for the high dose in females only. And there were no organ weight changes in the brain, adrenal, heart, kidney, spleen, thymus, testes, thyroid, ovaries, or uterus. And so we did see changes in liver weight. We expected to see that. And we did see changes. They were significant in males and females at 2250 and 4500. And we also saw hepatocyte hypertrophy. It was minimal and mild, and it was mainly centrolobular. So we saw this at a little bit lower doses. So we started to see it at the 750 ppm. But these findings are non-adverse adaptive changes. And then these are the metabolomics results. So here we saw, so there was some sample variability, but there was still a clear dose response in both sexes. And so we saw metabolomic changes in the liver. There were higher levels of cholesterol esters and long chain saturated triglycerides, lower levels of tocopherol, and shorter chain unsaturated triglycerides. And This occurred although there were no changes in the circulating cholesterol or triglyceride levels. Sorry. PPAR alpha seemed unlikely since there were no shifts in sphingolipid concentrations. There was a change in lipid profile in the liver, and that could potentially be due to direct effect on compound X mode of action. But we would have expected to see increasing triglyceride levels. The hepatic cholesterol ester transfer proteins and lipoprotein lipase expressions are regulated by LXRs, and we did see LXR pathway increased in the 14-day transcriptomic pathway analysis. There is still ongoing liver biochemistry work being conducted for this, and so that will be used to definitively determine if we did see CAR activation and also to rule out the other nuclear receptor activation. And there is a risk of liver tumors indicated to the project for dose levels excess of 2250 due to the magnitude of the liver weight changes. And so now this is all of the 90-day results compared to the 14-day BPODs. And at the bottom are the 90-day results. And here I took the liver histopath and did benchmark dose modeling on the 90-day liver histopath. This is non-adverse changes. So this would be considered a NOEL, not a NOAEL. And so these are pretty conservative. And as you can see, the one that is the most conservative is the female. And this aligns nicely with the BPODs that we're seeing. And I have another slide that separates it out so it's not so busy between male and female. So you can see in the male, the liver NOMAL, this is the metabolomic. And this is the 90-day liver histopath. And so you can see that the two BPODs, we had a liver and a bone marrow one, and they are both more conservative than what we saw as the apical effects. And then over in the female, this is the liver histopath, and this is the 90-day NOMAL. And also, we are seeing that these are in concordance with the adrenal, which was the lowest BPOD in the female. And so in summary, the 14-day transcriptomic point of departures, they provided good approximation of the toxicological potency of the most sensitive 90-day apical effects. The bone marrow was the most sensitive response in the male, and the adrenal was the most sensitive organ in the female. But the most sensitive 90-day apical effects were in the liver, and that was the central lobular hypertrophy. There were also effects seen. There were LDL and globulin findings, which were also considered non-adverse. And the study's NOAEL was 2250. So the transcriptomic point of departures were predicting the NOEL rather than the NOEL. And the 90-day metabolomic results showed concordance with the 14-day BPOD and pathway analysis. So for the next steps, so we have ongoing work to better understand the false discovery rate. SIOM created some null data sets for us and ran the parameters that I used to analyze this data. I've also been working with some other ways to analyze data. And so we're trying to understand what's the best way to analyze this data, and do we need to be concerned about the bone marrow and adrenal effects, or was this just a false discovery? We also have liver biochemistry work is ongoing, and the 2-year carcinogenicity study is underway. We'll be getting results back during the 2 years, so we don't have to wait all the way to the end. And we have several more chemicals that have undergone the 14-day transcriptomic analysis. They all have different responses. And so some of them, like the example I showed you today, showed dose proportionality, but we've seen some that haven't. And so, so for our next steps, we'd like to integrate the TK work into the BMD modeling, so that we can better understand what is the organ exposure, and to be able to relate those molecular and cellular changes to the organ exposure. We also have continued method development happening. We are working on different ways of analyzing the data. There's different methods. I've been working with Scott Auerbach and looking at this 3SD pre-filtered method, as well as just changing the changing different parameters, and just determining how to determine what is the best way to analyze the data set, and is there like a tiered approach to this? So do you look at a data set and say, since this data set looks like this, we'll choose this method to analyze it? Or do you set we analyze all data this exact way, and that's the only way? And so we also have been looking at multiple tissues with our studies, rather than just some of the typical liver and kidney organs. And so we haven't yet gotten enough 90-day study results back to determine whether or not looking at multiple tissues is going to be useful or not. And so then we'd also like to be looking at in vitro transcriptomics, as well as these in vivo transcriptomics. And so with that, I'd like to thank everybody that helped with this project. So over here is our scripts team. So Katie Bailey is the chair of that team. And so we have Katie, Jeff Davis, Liam, Amber, Caleb, Richard Curry, and David Cowie. And then I'd also like to thank Liz McInnes, Doug Wolf, Doug Brown, Laura Goldsworthy, and Brett Jeffries. And then also our collaborators. So we've worked with BioClavis, Harper, Ben Steinhaus, and also with several folks at Siome, so Rashir and Durrell. Also Scott Auerbach has helped me tremendously with analyzing the data, and we've had collaborations with Susan Hester. And we're always looking for new collaborators. So if anybody would be interested in working with us, we would be excited to collaborate. And with that, I'll take any questions.
Great, thank you, Dr. Stevens. Does anyone have any questions? Okay, I can start us off with one. So the results from the bone marrow, for instance, would suggest that that would be a target organ potentially in a longer term study. Does it make sense for that to occur with this known mode of action, for instance?
So so that was so I've done some null analysis work, and given our data set, we've made a few changes to the way we're analyzing the data. And so when I do add in that additional parameter, so that it reduces the false discovery rate to 5%, the bone marrow does disappear. So so I added that in here because I just didn't have time to really go through that that analysis. And so I think it's always important to keep the mode of action in mind and what you already know about the chemical, and does something make sense, and how, when you look at the data, does it look like this could be an artifact, or does it look like this could be a real effect? Yeah.
Okay, got it. So in other words, this could be an artifact depending on the way that you've analyzed it, for instance.
Yeah, but this was all very new information that just sort of came through. So before taking the bone marrow off for this presentation, I just decided to present it. But yeah, it doesn't. It's likely this one will jump off and will not need further analysis.
Okay. And so are there plans in the longer term study to also perform benchmark dose modeling on some of the potential target organs, or on a suite of tissues, as you mentioned?
So for the 90-day study, I believe samples were prepared in such a way that we can go back, but we, as of yet, do not plan on doing that work on the 90-day study. It can be done. So depending on what we see in our 2-year study, we can certainly go back and look.
Okay, great. Any other questions? Okay, great. Thank you, Tina. Okay, so our next speaker is Ken Elstein. He'll give a presentation on career development. So part of his presentation involves describing his career journey. So I will just provide an overview and he'll fill in some relevant gaps. So Mr. Elstein is currently retired from the EPA, where he was a widely published bench scientist when, after 18 years, he earned his MBA and did a career switch and pursued a career in organizational improvement at the EPA. He became a certified professional coach, a certified facilitator, certified administrator of the Myers-Briggs Personality Indicator and the DISC Behavioral Type Assessment, and a distinguished Toastmaster. And in retirement, he continues to coach individuals at all stages of their careers, including trainees and those considering options during retirement. He has several areas of specialization, and they include managing change and stress, time provide the resources tab, and we will send out a specific link regarding their specific location after this meeting. So with that, I will turn it over to you. So thank you, Mr. Elstein.
Okay, you're quite welcome. Thank you, Katie, and kudos to the steering committee for even thinking of putting a session like this in a science meeting. I think it's absolutely essential, of course, and it's interesting. I appreciate the introduction, and I will embellish it a little bit, but as much as I am advocating a strategic approach, let me get this slide working here, my career was anything but strategic. And I'm encapsulating a lot by saying floundering for the EPA. Basically, the way I have said it in various seminars and webinars and workshops and all that is, I grew up a nice Jewish boy on Long Island, New York, where basically there are only 2 legitimate careers as far as all the patriarchs and matriarchs were concerned, and they both end in the letter A. I'll let you ponder that for a second. Those who are from the Northeast probably get it. Down south, I mean, we're all in Durham, Raleigh area, may not, but it's a doctor or a lawyer. And I did not want to be a lawyer. And I wasn't too excited about the idea of being a physician, but I did like animals. So I decided I would be a veterinarian. And that was my target starting from age 14. And I just adhered to that and continued. The fact that it was very difficult to get into vet school just tempted me all the more. And so it was a big journey, and it actually included undergrad at SUNY Binghamton. So that's kind of cool, Logan. I didn't think we had any alumni out there. And ultimately getting into vet school, but I did not like it at all. And by the senior year, with the idea of doing this for the rest of my life, that I was just so opposed to that I dropped out, much to the chagrin of a lot of people. And so that's where you get floundering. Now, in that journey, I did have a lot of experience in labs. That's part of why, the kind of things you do to qualify or to get accepted to vet school. So that did allow me to get a job initially as a contractor to EPA, but then as EPA, but I never had the passion to really pursue science like all of you folks do. So as Katie mentioned, I went and got an MBA. That was actually the advice of a manager. Thought I was set up for management, thought I'd be a good manager. So I went in that direction, but then the company got taken over, a hostility by another company. And then there was a conversion, and that's how I became an EPA employee. So all these changes are going on that I never accounted for. But I did start exploring, figuring out what else do I want to do? And I went on to the administrative side, got into organizational improvement and ultimately leadership development, and then ended up settling on organizational development, which can mean a lot of things to a lot of people. But this is how I empirically determined it, by just trying to meet the needs that I was encountered. I didn't start doing these 5 areas, but they sort of evolved that way. And really, it's been the most rewarding part of my entire career. And yet, I think an important thing to recognize from the circuitous loop over here of a path was that when I was down here, this was never on my radar. In fact, even when someone mentioned coaching to me in 2003, I thought they were crazy when they thought I'd be a good coach. I didn't see it at all, but I tried it and I ended up loving it. And a lot of my clients are happy with what I can offer. And I appreciate that that Katie mentioned those booklets. I hope you folks will take a gander at them, because that does contain the information from not only my coaching, but my consulting and training experience as well. So what does this all come down to? I am also a reductionist by nature. I like to give simple messages. I think there's a lot of clarity in keeping things simple, not simplistic, but simple. And the 2 points that I think are absolutely important, and they're blended, is that you have a basic strategic planning view for your career, but you also honor the scientific method. So for some people, when you mention strategic planning, you get lots of eye rolls, and people involved in it will give lots of eye rolls as well. But it's not all that complicated, or at least shouldn't be. It's basically ensuring that you characterize the current state comprehensively. You characterize the desired state so you can figure out a path from current to desired, and then you, of course, assess your progress so you can adjust as necessary. You don't want to blindly follow the same path if all these other things, other variables come in and derail things. Or if you just find out that, like I did, that, hey, that path wasn't exactly what I wanted. I don't have to tell you what the scientific method is, but one of the things that I personally find amusing is irony. I think it kind of underscores the human comedy. And one of the biggest ironies that I have noticed is with my clients, who most of whom are scientists and engineers, is while they take pride in applying the scientific method in the lab and in the field, and they really feel that you should make no decisions unless you have the data to support it, and you should base your decisions on data rather than a priori. When it comes to their life, they don't do that. They will do their biases, other people's advice, whether it's well-founded or not, suggestions, opinions, and even fears. They won't go in an area because they're scared about not being perfect. Well, how are you ever going to learn something new, go on a learning curve, if you're terrified of screwing up? So my point is, keep a basic strategic planning profile in mind, but you have to experiment to learn what you like, as well as even talents you have that you didn't realize you had. So how do you characterize the current state? Basically, you should be able to answer these questions. And while you may have heard, perhaps in a performance review, that it's important to be aware of your weaknesses and vulnerabilities, I think a lot of people aren't even aware of their strengths and their talents. We can have blind spots to our talents because they come so naturally to us, we assume everybody can do it, but that's not the case. So to be able to have a comprehensive profile, what I would call a brand of everything that you can offer the world, and of course it'll be growing over time, is absolutely essential. So how do you go about figuring that out? There are lots of ways. I mentioned this brand statement. Actually, there are two brand statements, and this is all in one of the handouts. Finishing first is the name of the handout. So don't worry if you're not catching all of this. The core brand is your passions and the totality that you bring to their pursuit. And again, it's got to be comprehensive. I'll talk more about that a little bit later. When it comes to applying for a job, there's something else. I call it the anchor brand because it is anchored to the vacancy announcement. And the bottom line for that is you are trying to make the convincing case that you are the best person for the job. You do, in both cases, you have to show both aptitude here, not KSAs and the strengths and the talents, but also attitude, the passions that you bring to it. So people know that you will go above and beyond. When it comes to the core, you might not even be clear what you want to do, but you have an understanding of what you really care most about. And that could be because it's something that's existing that you want to keep moving forward, or it's something that doesn't exist and it actually angers you. So for example, people who feel there are environmental injustices and they're pursuing environmental justice, they come from a true passion of something that currently there's a lot of inequities. Also, you have to understand what do you really care about. And there's this tool called a Wheel of Life. I'll mention it shortly, but it just helps you to be conscious of it. We cannot be everything to everybody, but if you can consciously make those trade-offs and be satisfied with it, then you can come up with something that you can truly aspire to. And you're not going to be satisfied in life unless you know what you're shooting for and you're making progress to it. If that target is kind of abstract or diffuse or abstruse, then you're always going to feel like you're wanting something. It's never good enough. So it's important to clarify, what do you really care about? There are plenty of assessments out there to help you figure out your current state. There are online assessments, and in the handout that's associated with this webinar, there's several little tools and exercises you can do to understand better. You probably have heard of the various personality types like Myers-Briggs. There's actually quite a lot of information in that handout about that. Or the DISC, or values, psychological needs. You certainly, hopefully, will get meaningful performance evaluations that will give you a sense of not only where you didn't do great, unfortunately, and many managers like to focus on that, but what you did do very well, what your skills and talents are. And then there's this concept of high point stories that also is discussed in the finishing first handout, and actually both handouts. And that's to reflect on the times that you were at your best. It doesn't necessarily mean you had you won a major award, or you hit a grand slam home run, but it's something that was really important to you, whether it's at work or in life in general, because buried within that are factors that have allowed you to succeed, as well as your passions and your interests. The Wheel of Life is not a 10-year-old girl doing a cartwheel. It's this little tool here. And really, these are suggested. I got this online, but you can change the various parameters if you want. The point is, as I said before, to make the conscious decision of how do you feel you're doing relative to what you'd want, not what other people would want, but what you would want. And so I just put a few X's up there, and then you kind of connect it and see, how balanced is that? So in this scenario, this person is pretty happy. They're healthy and they are happy with the level of family and friend support, and they have a nice relationship, and they're happy where they live, but they're not so happy where their career is going. And as much as they're putting a lot of energy, they don't feel that they're getting much growth, and yet they don't have any time for fun and recreation. And so taking the moment to just make that assessment is useful. Now, upon reflection, they may realize with the trade-offs that while they don't have as much time for fun and recreation as they would like, given the possibilities, it's a sacrifice they're willing to make, at which point they might regrade this to be something more like, an 8. The point is, just take the time to figure this out so you know what you want, and therefore you know when you hit it, you'll know when you hit it. As far as the desired state, obviously that Wheel of Life will help there as well, but you do need to know where you're going, otherwise you'll be floundering like I was. Now, it doesn't mean that you have to have that in concrete right now. It's at this point, what do you think your desired state is? So these are the kinds of questions you would try to answer. And as far as how to go about doing that, again, I mentioned the high point stories. I do that a lot with clients. By the way, this isn't a commercial for coaching. I'm just offering. If people are interested in having some conversations, I'll be glad to do that, but I'm not advocating. It's not an infomercial or anything. But I use the high point stories. I find that people often undervalue themselves. And again, they don't often realize that the things they've done are actually impressive just because they didn't think it was a big deal. But I also advocate networking. You really need to talk to people, find out about their career journeys, connect with your tribe if you want to change your career. I've had clients that have wanted to move into sustainability, especially during the last administration, where that wasn't exactly valued too much by the administration, but they were able to find their tribe and make some connections and even get some job opportunities. Mentoring, coaching, I'll talk about that a little bit more. But again, with that other major point here about being a scientist, it's really important to experiment. You have to get out of your comfort zone into a stretch zone, but you don't want to be in a panic zone. So you have to kind of navigate that. But also, as a scientist, you have to analyze data. When you do these things, when you stretch yourself, you have to look at what did you learn from that. I profess and I encourage people to use what I call the plus delta reflection. Don't focus on what went wrong. Nobody's perfect. And focusing on your screw ups is not going to allow you to move forward. But you do the plus delta, the pluses, what went well that you want to continue to do and even build upon. The deltas, even for things that didn't go well, what do you know now? You didn't know before. And based on that, what are you going to try to do next time, your next experiment? As far as clarifying a path, you may have heard the concept of a SMART goals. It's just the point of being very specific rather than being vague that, you know, I want to earn more money sometime. You want to get more specific about that. How much money do you want to earn by when? And you might even want to do a financial analysis, especially if you're going to prepare for retirement. Trust me, folks, I'm retired. Financial analysis is important. So you want to get into specific goals. Some SMART acronyms use specific and the M is measurable. I like another mile that says motivated. You should be excited about that goal. It should certainly be achievable. Don't frustrate yourself.
Hello.
Yes, looks like we're having technical difficulties. Everybody please hold. Try to get Ken back on the line. Ken, you're on mute. You're on mute, Ken.
Hey there, we good?
I hear you now.
Okay,
Yep.
Figures, you know, I didn't lose connectivity all this time and now. All right, so to understand the path and even possibly the dependencies, what experiences, what positions, what certifications you may need, it helps to have those conversations. And of course, you're building your network throughout the whole thing as well. You want to build that collection of advocates who will expose you to opportunities and be champions for you. But you can, again, you have to set priorities in all of this, and that's going to be an important part of it. Ultimately, though, it's going to come down to what are you going to do by when, those specific actions. Otherwise, you'll never make progress to it. With regard to helping, there's lots of people. Hopefully your supervisor will help. I've definitely worked with clients who didn't have very supportive supervisors, but that's why you want to go out and have these other conversations. They might help with an individual development plan, which does this more formally. It's almost like a discussion instrument. You clarify what your goals are and you offer some various options, and then you have this sort of negotiation discussion as to which they'll support, especially if money is involved, and or even time. You need time away from some of your duties to do a detail, for example. Again, I'll mention coaches, mentors shortly. Also, it's really important to have very good interviewing skills and networking skills. This again comes down to branding. There are a lot of people who, clients I've had, who don't get jobs, not because they're not qualified, but because they don't know how to convey their qualifications in a compelling way. So helping them build their interviewing skills, helping them put their best foot forward in their application package is incredibly important. Same thing when you're having these conversations. You want to be able to convey very clear to the other party what you're interested, what you bring to that, and what you need from them very succinctly, because these can be very busy people. And if they feel you're wasting their time, that'll leave a bad impression. So you want to be able to have all this resonant in your mind and even have your 10-second cold call introduction, your 30-second introduction. But also essential soft skills. I've kind of gone in and out today, and it looks like you've got some really great presentations. These are great presentations to a technical audience, and a lot of scientists are really good at that, but when it comes to a non-technical audience, they flounder. And I'm not talking about an outreach to a high school. I'm talking about your senior management when you're trying to convey why they should fund you. If you can't get to the point really quickly, then you can lose them. The handout about getting to the point came because the Air Office asked me to train their scientists, because the administrator, this is EPA, the administrator would want a briefing. The scientists would hand them a handout of 100 slides to go through this briefing, and the administrator, when they're on slide 1, the scientists on slide 1, the administrator, on slide 63, you say, oh no, wait, I'll get to it. No, you don't do that. You get to slide 63, and you don't give them 100 slides to begin with. Then you also have to make these compelling proposals. Why should they invest in you versus some of the other people? Interpersonal skills, especially, the more we get into interdisciplinary approaches, the more we have to interact effectively with people outside our discipline. So the ability to get along with others is incredibly important. If you want to differentiate yourself moving up, communication, influencing skills, and personal skills are extremely important throughout that entire process. Brain management will be very important. That is not only being able to effectively manage other people's brains, keep them from going ballistic, but even managing your own, not letting other people adversely affect your self-esteem, and even not allowing the stress of the moment to derail you. The brains work very well under moderate stress, but horribly under high stress. So it's important for us to be in a situation where we are managing our stress response. And obviously there are also psychological and health consequences of always being under high stress. So the more that we understand that, the better. There's a handout that goes into the frameworks and models that I use on that. That's another big area that I coach people in. But I'll reiterate it to the point of being a dead horse, but you have to experiment. You have to do those stretch assignments. And that can be at work, and even be outside. You can do volunteers for community or other societies, and doing an officership in a society like GEMS, whatever. Try these different things. You may not like it, but that's important to know. But on the other side, what you're going to be doing over time is building your pedigree, expanding your resume, but also your network, and you will be able to use that as you move forward. One of the things that I advised for my EPA people, there was a lot of happiness at the election that we now have a very supportive administration, and that's great, but there are no guarantees it'll stay that way. So I've been telling people, what's the resume and the network you want to have in 2024 if the circumstances change such that you would like to have a job outside government, or maybe just even outside the agency. It's something worth thinking, and that is strategic, because you start thinking about, where do I want to go and what's the path to it? About brand. So everybody on this call, I'm sure, can compete on specialized knowledge, their various skills, data analytics and project management, maybe a couple more. But I have clients who come in, and it's almost like they think they can compete with everybody else on just one or two dimensions. And there's always going to be somebody who's got more publications, more experience. What you really have to do in being comprehensive is start thinking the other things. And again, it's like high point stories and other things that allow you to recognize whether you have these, and don't be your own worst enemy in the sense of raising the bar. If you've done something once, you can be an expert depending on what it is. That's not true in the sciences, but it can be true in strategic planning and in conflict management and a few other things. So going through that exercise to make sure that you are being fair with yourself is really important. And it actually will bolster self-esteem. I have had clients that because they were in a situation where it wasn't a very good fit, they had a crisis of self-esteem. In fact, I had one client who, to hear, you would think this person was a polymath, a Renaissance woman, a physician, an artist, an actor. She even met Robert Redford when she was skiing. But it was, she was in a situation where, from even a Myers-Briggs personality type standpoint, it was a really bad fit. She kind of got baited and switched, or she got moved to a position that wasn't what she applied for. And she was crashing. Health-wise, she had gained weight, blood pressure was up, she was pre-diabetic, pre-hypertensive, and it was looking really bad. Her pastor, her doctor, everybody told her that if she stays on this, she's gonna die. And so she came to me, we worked on her brand, and then she did networking at a level that I could not possibly imagine anyone doing. She was networking with CEOs and administrators. I mean, she was at least confident. And she always got the message, my God, if I had money, I would scoop you up. Because at that time, especially, this was the crisis, the recession. And so that nobody had any money. But it was really great for her to get those messages that, hey, it's not you, it's the position you're in. So just an example, but you have to figure that out. And then you use that again as core brand and as the anchor brand. Who can help you succeed? Let me just, how are we doing on time? Who can best help you succeed? Mentors within the organization, often they have experience in the job you want, or they're connected so they can give you advice, expertise, etc. Life coaches will not necessarily have the expertise, but they'll bring in the personal aspect of it and try to help you understand and think through the goals and problem solving and all that. There are people who operate in the middle. I do, the expertise I offer is what's in the handouts. For people at an organization, they might move into this area. I've trained mentors at an EPA that move into this area, but they're unlikely to go into the personal side of it. And you can understand that that might be uncomfortable, possibly even inappropriate. But if the personal side gets to the point where people just can't move forward, they need to resolve something, then it's important that they seek professional health, from a mental professional. From the progress, it's just a matter of keeping eye on where you're going and assessing. Again, you're not using this as a way to pass judgment on yourself. It's more, like I said, with the plus delta, what are you learning? And you have to expect that there will be bumps in the road. Nothing is going to go smoothly. To me, the only failure is when you give up at a time when you shouldn't. I mean, it wasn't a failure for me to give up on vet school because that was a bad fit. But if I try something, it didn't work and I give up, then that's way too premature. I think a really important additional consideration that especially social media is really underscored is reputation management. And that's hugely important in any organization. We tend to get concerned, appropriately enough, when someone is attacking us. We have to be cautious about whether we respond to it or not, because the brain is already pre-programmed through evolution to be attracted to the negative stuff and to adhere tenaciously to it. So if somebody creates a negative image, it's very hard, if not impossible, to negate that negative image. We have to replace it with a positive image. And so we have to be careful about defending ourselves if it's reinforcing a negative image. As the saying goes, if you try to prove that you're not crazy, you'll probably sound crazy. So instead, what's really important, I think, is to be proactive. Like I say, again, build that network, develop that a network of champions, allies, and advocates who will get you opportunities. Identify those people who have influence and try to connect with them, do work, do a briefing for them, whatever, so that you get their attention. I'll just reiterate again, it's this whole idea of incremental empiricism. Any situation, whether it's your career, trying to figure out where you want to go, or you're dealing with a conflict with somebody, just take this approach. Try to understand the current situation and clarify what you want. What's the desired state in this relationship? If you're in a conflict, you may feel like, all right, where is this person coming from? And the brain management information, the intent there is sense making, trying to understand what might at first blush appear like nonsensical behavior, but why might that make sense to that person? Where are they coming from? Have that empathy to try to get inside their head. So then you can do an experiment of approaching them differently. Rather than a fight or flight approach, try to do something that's a little bit more interactive, a little bit more of what they call emotional intelligence. And then do it. And if it works, great. Why did it work? What can you carry forward? If it didn't, what are you going to try to do the next time? And again, that plus delta assessment will help you do that. With the free booklets, the first one is supporting what I've covered today. Of the others, I would say the two that I end up using the most, again, Finishing First, that lays out how to find a job from the very beginning all the way to negotiating your compensation package. And then this other one about, that's brain management, exerting influence in your world, understanding how the brain works, especially based on evolution. By default, we tend to have behaviors that are self-destructive, and we need to have that awareness so that we can be more constructive. And with that, I'm happy to take any questions. Also, my email is down here. It's also on the second page of all the booklets. So I'll be happy to answer any questions that you want to send my way. But just think about these. Again, I think it's fantastic that GEMS is reaching out to do this and that they are really interested in helping you develop your career. So I'll kick it back to Katie or George or whoever wants to wrap this up.
Yeah, great. Thank you so much, Ken, for the great presentation. And just to mention, like initially, we're going to send out a survey soon that will sort of ask you some things that you're looking for in terms of career development. So please look for it when it comes out. But any questions at this point? Sure, Caitlin.
Hey, my name is Caitlin. I don't really have a question, but I did want to say thank you for coming and doing this. I am new to the GEMS team, and I am also a current graduate student, and I really appreciated a lot of the insight that you were able to give. So thank you for being here.
Oh, you're quite welcome. I'm glad it registered, resonated.
Amy?
Hi, I'm Amy Wong. I used to be a postdoc at EPA.
Yeah.
And I remember you were very eager to help people. I probably threw you one of the questions that you never anticipated when I was a postdoc. I was pregnant at the time, and I really wanted to know how to do professional while having a big belly. And you delivered. I couldn't believe. I was hoping you know somebody who are good dressers and could share experience, but you actually found a brand that has a local shop, and I shop there. And I have to say, I look great during my whole pregnancy.
Well, thank you. I wish I could remember that. I'm glad you do. That's the other thing when you had retirement, your memory fades. But thank you very much for that.
I do have a question, is
Sure.
that sometimes when we move from position to position, you lost the bridge, you lost connections, or even you felt guilty about not doing something as well as you had hoped in your prior assignments or projects. Are there ways you can recommend how to rebuild those bridges? No.
You're asking a simple question. I'm trying to give you a simple answer because it's a little bit complicated. I never think there's a wrong time to rebuild a bridge. I think what it comes down to is how do you make that segue, or how do you connect to it, or reconnect to somebody. I'll tell you this. So, all right, here's a tool. It gets into the brain management stuff. And it's what I would say is my Pareto thing, the 20% that'll get you 80% of the way there. In any communication that you want with somebody, there are 3 elements that I would include. 1 is to clarify your intent. Why are you reaching out to them? because the brains being what they are, if we're not clarifying our intent, and it should be outwardly focused, you're trying to help a larger cause or help somebody else. If it isn't that, people will assume it's self-interest and they get turned off. So you want to be able to convey an aspiration as to why you're reconnecting. The other piece, which in your case you might actually do first, is appreciation. You connect with the person and say something sincere about them that you sincerely appreciate. And even if you didn't have a great experience with them, you can still say what you admired about them. And then the last piece you end with is a question, it's inquiry. The reason is that it values the other party because you're asking for their opinion. It gives them control of the conversation so that they have some say as to how to move forward. So what does that look like? Amy, we haven't talked to each other for 10 years, but I always found your advice to be incredibly valuable, and I've definitely respected your experience. And I'm at a point in my career where I'm considering changing my profession, and I would really appreciate a few moments of your time to talk about, to get your thoughts. You know me. I'd also like to update you on where I've been since then. Would it be okay? Is there a time that we could set up even for a 15-minute conversation? Maybe I could treat you to a cup of coffee. And the other piece of that, which gets to branding, is you're going to come to that conversation with your brand, the totality of what you offer now, not what you offered 10 years ago when you were making mistakes. Does that help at all? Like I said, it's a kind of question that's best answered with a customized answer, but that's my shot by way of a generic response.
It's wonderful. Thank you.
Sure. By the way, when people ask the time, I tend to tell them how to build a watch. So just so you know.
Great. Well, thank you so much. We really appreciate you coming in. I just want to mention that someone put in the chat from Logan Everett, Ken, don't forget about a famous Binghamton alum, Paul Reiser. So just want to make sure you saw that.
Oh, really? Okay. Thanks, Logan.
Okay, so this just about wraps up our program. Before I turn it over to George for some closing remarks, I just wanted to say my sincere appreciation for everyone to join, especially all of the speakers for sharing their work and insight with us. It's very much appreciated. Thank you. George?
Thank you guys for organizing this, your whole steering committee.
All right, well, exactly. You stole my thunder there, Ken, because I also wanted to thank the board of directors, especially you, Katie, and for leading the planning committee. And the planning committee consisted also of Laura Taylor, Amy Wong, Caitlin Meyer, and Leah Wehmas. So thank you to everyone. Now, I also have a few general announcements that I'd like to make. First, Katie mentioned in passing a survey that's coming out to GEMS members and also contacts. We're going to be looking for feedback on the new GEMS website. We're also going to be looking for your input on scientific topics for future meetings and/or webinars. We're also looking at developing more along the lines of career development, much along the lines of what Ken had been presenting. And there's other activities and services that GEMS has been providing and maybe could provide for other members. So your input on all of those different aspects is being sought out through this survey. Next, I also wanted to encourage you to get involved. And Ken mentioned that briefly in his discussion. There's a lot that you can gain from a lot of these outside activities that are not necessarily your day-to-day job, but they still help to advance your career. In GEMS in particular, we have a number of subcommittees that we're trying to stand up or re-stand up. One is a website committee. Laura Taylor is leading that. We also have an election nomination committee, and Carol Swartz is going to be heading that. I'll speak a little bit more about that in a moment. We also have outreach, and with that, we hope to institute a newsletter that'll come out on occasion. And then a social committee, and Caitlin Meyer will be leading that effort. Now, going back to elections, we do have upcoming elections for the board of directors. We will be looking for candidates, and we'd like for you to consider either volunteering yourself or nominating someone else. Stay tuned. More details will be coming in coming weeks. And then lastly, we have our fall meeting, which is planned to be held in person at the North Carolina Biotechnology Center. That's typically where we've had it for the last few years. When we have been able to meet in person, we're looking at the first week in November, and the scientific theme will be duplex sequencing. Details again will be provided in the GEMS website and in emails that will be forthcoming, and hopefully a newsletter that will also be coming out sometime in the coming year. So with that, thank you all for attending. I think this is a very good and interactive meeting. Appreciate everyone taking the time. We had upwards of almost 60 participants in this meeting. So until next time, take care, be safe.
Thanks, George. Bye-bye.
Bye.